There is a version of this planet that no human being has ever seen. No photograph captures it, no painting comes close. No dream has ever conjured it accurately, because the human imagination was built inside this world, the familiar one, the green and blue one, and it simply does not have the vocabulary for what came before. Go back far enough, and Earth becomes a stranger. 3 billion years ago, if you were somehow standing on the surface of this planet, the first thing that would kill you is the thing you cannot see. Not a predator, not a fall, not the heat, though the heat would be extraordinary. What would kill you is the air itself, or rather, the complete absence of the one ingredient that your body has been built over millions of years of evolution to require absolutely. There is no oxygen here, not a trace. The atmosphere pressing down on this alien landscape is a thick, chemical soup of methane, carbon dioxide, nitrogen, and hydrogen sulfide gases that your lungs would process for approximately 30 seconds before the signals from your brain stopped making sense. But look past your own survival for a moment. Look at the world itself. The sky above this ancient Earth is not blue. It burns in permanent shades of orange and deep rust red, coloured by the methane haze that drapes the entire planet like a chemical veil. The sun hangs in this strange sky, but it is not the sun, you know. It is younger, dimmer, about 70% of the brightness it carries today. And yet the planet is warm, held together by a greenhouse blanket so thick that even a weakened sun can maintain liquid oceans across the surface. Those oceans do not look like any ocean you have ever seen. They shimmer with a faint chemical iridescence, traces of iron, sulfur compounds, dissolved minerals that would be toxic to every marine organism alive today, swirling through water that has never once felt the pull of a tide created by something swimming beneath its surface. Nothing swims here that is large enough to see. Nothing walks the barren, rocky shorelines. The land is raw and dark volcanic rock, ancient basalt, no soil because soil requires organisms to create it, and no organisms capable of building soil have yet arrived. This is Earth, the same rocky body, the same orbit, the same moon pulling at the same tides, but completely, utterly alien. And here is what matters. Here is what this story is actually about. This world, this oxygen-starved, methane-choked, chemically hostile world, is not empty. It is, in its own way, extraordinarily alive. And something living inside it is about to change everything. The rulers of this ancient world are invisible. They have no eyes, no mouths, no limbs, no voices. They leave no footprints on the volcanic rock. They build no structures visible from any distance. And yet they are everywhere in the oceans, in the sediment, in the deep cracks of the seafloor, in the scalding water pouring from hydrothermal vents at temperatures that would dissolve human flesh. They are, in every meaningful sense of the word, the dominant life form on Earth. They have been the dominant life form on Earth for longer than the human mind can honestly comprehend. And they have built something remarkable, a living world perfectly, elegantly, completely tuned to the absence of oxygen. These organisms are bacteria and archaea, single-celled, microscopic, ancient beyond any intuitive reckoning. They breathe sulfur instead of oxygen. They consume iron. They digest methane and release carbon dioxide. They extract energy from chemical reactions that would seem impossibly exotic to any modern biologist trained in the oxygen-rich world we inhabit. But to them, these reactions are simply life. This is simply how the world works. This is how the world has always worked. And the world, by any objective measure, works beautifully. In the deep ocean, vast bacterial mats carpet the seafloor in living tapestries of orange and purple and deep crimson colours produced by the pigments of organisms that have never needed to photosynthesize in the way a plant does, that draw their energy from the chemical gradients between hot volcanic rock and cold ocean water. Near the hydrothermal vents, communities of microorganisms thrive at temperatures that would destroy every protein in a human cell. In the shallower waters, where a dim sun filters through the chemical haze, purple and green sulfur bacteria use light to drive their own form of photosynthesis, but a form that releases no oxygen, a form perfectly calibrated to leave the atmosphere exactly as it has always been. The chemical cycles of this world are in perfect interlocking balance. Methanogens produce methane, which warms the planet. Sulfur-reducing bacteria process sulfur compounds and release hydrogen sulfide, which feeds other organisms. Iron-reducing microbes strip electrons from iron oxides and release ferrous iron back into solution, where it becomes food for yet another set of organisms. Every atom cycles through this living system in a loop that has been running, without interruption, for over 1 billion years. This is not a primitive world. Do not make that mistake. This is a sophisticated world, a world that has had 1 billion years longer than our own to refine and perfect itself. The organisms living here are not failures waiting to be replaced by something better. They are champions. They are the most successful life forms this planet has ever produced. And they have absolutely no idea that one of their own distant relatives, multiplying quietly in the sunlit shallows somewhere above them, has already begun to end their world. In the shallow margins of the ancient ocean, where the dim light of a young sun still managed to penetrate a few meters of murky water, something new had appeared. It was small, smaller than small, really, a fraction of the width of a human hair, invisible to any eye that would not exist for another 2 billion years. It looked, under any sufficiently powerful microscope, unremarkable. Just another bacterium. Just another single-celled organism drifting in warm, mineral-rich water, going about the business of staying alive. But this organism had a secret. It had discovered, through the blind and patient trial of evolutionary accident, a way to use sunlight that nothing on Earth had ever managed before. Not just to drive simple chemical reactions, not just to shuffle electrons between sulfur compounds, but to do something far more radical to split water molecules apart. Hydrogen from one side, oxygen from the other, using the captured energy of sunlight to power the separation and harvesting the hydrogen for its own metabolic needs. The hydrogen was the goal. The oxygen was the accident. These organisms, what we now call cyanobacteria, though they had no name then and needed none, were not trying to change the world. They were not aware that they were doing anything unusual. They were simply following their biochemistry, as every living thing does, extracting energy in the most efficient way their particular molecular machinery allowed. The oxygen they released with every split water molecule was, to them, metabolic waste. A byproduct. Something to be discarded. They discarded it into the ocean. And for an extraordinarily long time, millions of years, then tens of millions of years, nothing seemed to happen. Because the ancient ocean had a defense. Dissolved iron, present in vast quantities throughout the primordial seawater, reacted eagerly with the released oxygen, combining to form iron oxide rust, which then sank to the seafloor in a slow, continuous rain of reddish particles. The ocean was acting as a chemical sponge, absorbing every molecule of oxygen as fast as the cyanobacteria could produce it, locking it away in solid mineral form before it could reach the atmosphere. On the seafloor, these iron oxide particles were accumulating into something extraordinary. Layer after layer of rust-red iron oxide, alternating with layers of dark silica-rich sediment, building up in rhythmic geological strata that would eventually solidify into some of the most ancient rock formations on Earth. Today, we call them banded iron formations, and we mine them on every continent for the iron in our steel and the ore in our industry. Every skyscraper, every bridge, every ship, built, ultimately, from the chemical byproduct of a reaction that was already loading a planetary catastrophe 2.5 billion years ago. But iron, however abundant, is finite. And the cyanobacteria were not stopping. They were multiplying. They were spreading across every shallow, sunlit surface of every ocean on the planet, splitting water molecules by the trillion, by the quadrillion, by numbers so large they dissolve into pure abstraction. The oxygen kept coming. The iron kept absorbing it. The layers on the seafloor kept building. And somewhere, in some stretch of ancient ocean that no map will ever locate, the last free iron atom was finally consumed. The sponge was full, and everything that came next was inevitable. There is a specific kind of silence that comes just before something breaks. A structural silence, the kind a bridge makes in the last fraction of a second before the metal gives way. The kind a dam makes when the water has reached the final inch of its capacity. The ancient ocean had been holding for hundreds of millions of years. It had been doing its chemical work faithfully, absorbing oxygen as fast as it arrived, sequestering it in iron compounds on the seafloor, maintaining the atmospheric status quo that every living thing on the planet depended on, without knowing it depended on anything at all. And then it stopped. There was no explosion, no earthquake, no visible signal of any kind. Just a quiet, invisible chemical threshold crossed somewhere in the sunlit waters of an ancient sea, the moment when the last accessible iron had been oxidized, when the last molecular handshake between iron and oxygen had been completed, when the sponge that had been absorbing the byproduct of cyanobacterial metabolism for hundreds of millions of years was simply, completely, irreversibly full. After that moment, the oxygen had nowhere to go but up. It rose through the water column and crossed the surface boundary into the atmosphere slowly at first, in concentrations too small to measure with anything but the most sensitive geological instruments, but steadily, relentlessly, with the blind persistence of a process that had no reason to stop and no mechanism to slow itself down. The cyanobacteria kept photosynthesizing, the water molecules kept splitting, the oxygen kept coming. Geologists call what happened next the Great Oxidation Event, and the name is both entirely accurate and somehow insufficient for what it represents. Event suggests something that happens and is done a moment, a flash, a discrete occurrence. What the Great Oxidation Event actually was is better described as a planetary transformation, a slow, irreversible chemical rewriting of an entire world that took somewhere between 200 and 300 million years to complete. But that was, on the scale of Earth's 4.5 billion year history, almost shockingly rapid. The evidence for this transition is written in stone, literally. Ancient sedimentary layers from around 2.4 billion years ago show the abrupt disappearance of certain minerals that can only form in oxygen-free environments. Uranium deposits, pyrite crystals, certain iron-bearing minerals that dissolve or oxidize the moment oxygen reaches them. They vanish from the geological record in a geological instant, replaced by the oxidized minerals that can only exist in an oxygenated world. The chemical fingerprint of the transition is unmistakable, and it appears in ancient rock formations on every continent on Earth, all pointing to the same extraordinary moment in planetary chemistry. The world was changing. Not because anything had decided to change it, not because there was a plan or a purpose or a direction to what was happening, but because a microscopic organism had stumbled onto a chemical reaction 2 billion years before anything existed that could appreciate the consequences. Something new was in the air, and for nearly everything alive on this planet at that moment, that new thing was going to be the last thing they ever encountered. We have been taught, from the very first moment we learned about science, that oxygen is the molecule of life. We breathe it. We need it. Without it, a human brain loses consciousness in 4 minutes and dies in 10. Every textbook, every biology class, every documentary about the natural world presents oxygen as the gift, the essential ingredient, without which nothing interesting happens on a planet. But that story is told from inside the oxygen age. It is the story of a world that has already been transformed, already adapted, already built around the presence of this molecule. It is not the story of the world that existed before the transformation. To the organisms of the ancient anaerobic Earth, oxygen was not life. It was the most destructive substance that had ever entered their environment. And the reason for that destruction is written in the chemistry of the molecule itself. Oxygen is, at its core, a molecular thief. It carries an urgent, almost violent electrochemical hunger, a need to pull electrons away from other molecules, to grab and bind and react with whatever it touches. In a world that had been built entirely around oxygen's absence, this hunger was catastrophic. Every enzyme, every protein, every lipid membrane, every metabolic pathway in every anaerobic cell had been constructed over 1 billion years of evolution without any need to defend against oxidation, because oxidation had simply never existed. These organisms had no armor against it, no antioxidant systems, no repair mechanisms for oxidative damage, no evolutionary memory of a threat they had never encountered. Oxygen hit them like fire hits paper. The chemistry of what happened inside these ancient cells is, in a sense, identical to what happens when metal rusts or when a cut apple turns brown oxidation. The aggressive combination of oxygen with other molecules, breaking bonds and destroying structures that had been operating perfectly before the intruder arrived. Inside an anaerobic cell, this meant the destruction of key enzymes that drove metabolism, the disruption of electron transport chains that generated energy, the oxidation of iron-sulfur clusters that served as the molecular engines of countless biochemical processes. The cell's own chemistry began to fall apart, not because anything had attacked it from outside, but because the environment itself had become lethal. And here is what makes this story genuinely terrible, in the oldest and most profound sense of that word. These organisms had done nothing wrong. They had committed no evolutionary error. They had not failed to adapt to anything that had ever existed before. They were the products of 1 billion years of extraordinarily successful biological refinement organisms, so well suited to their world that they had dominated it without competition for longer than any life form since. They were destroyed by a gas that had never existed in their world, produced by a relative they could not perceive, as the accidental byproduct of a reaction they had no way to influence or prevent. The invisible poison spread through the ancient oceans in slow, spreading plumes, and what it found in its path was a world that had never once, in its entire existence, needed to imagine that anything like this could happen. It had no time to learn. Ask almost anyone with a passing interest in natural history to name the greatest mass extinction in Earth's history, and they will almost certainly give you the same answer: the asteroid, the one that struck the Yucatan Peninsula 66 million years ago and ended the age of non-avian dinosaurs. It is the extinction we teach, the one we film, the one that fills natural history museums with dramatic skeleton reconstructions and inspires blockbuster films about prehistoric giants brought back from the amber. It killed approximately 75% of all species on Earth. It is, by any ordinary measure, an almost incomprehensible catastrophe. But it is not the largest extinction in Earth's history. It is not even close. The Great Oxidation Event, unfolding over hundreds of millions of years, beginning around 2.4 billion years ago, did not kill 75% of life on Earth. It killed, by the most conservative scientific estimates, something closer to 99%. Not of species in the modern sense, because species diversity in a microbial world is measured differently and more uncertainly, but of the actual living biomass, of the dominant biological lineages, of the metabolic strategies and ecological communities that had defined life on this planet for over 1 billion years. The vast majority of it was either destroyed or driven into the most extreme and inaccessible corners of the Earth, where it could survive only in the margins. And yet there are no museums dedicated to this extinction, no blockbuster films, no dramatic skeleton displays. There are several reasons for this silence, and they are all worth examining. The first is scale. Not the scale of the event, which was incomparably larger than anything that followed it, but the scale of time. 2.5 billion years sits so far beyond the reach of any human intuition of duration that it simply doesn't feel real in the way that 66 million years does. If you compress the entire history of Earth into a single 24-hour day, the dinosaur extinction happens at approximately 11:30 at night. The Great Oxidation Event happens around 9 in the morning. We are creatures of the evening, and the morning is simply too far away to feel. The second reason is visibility. The organisms that died in the Great Oxidation Event left no dramatic fossils, no skeletons, no footprints, no eggs. The evidence of their existence and their destruction is written in chemical signatures, isotope ratios, and mineral transitions in ancient rock, a language that required the development of modern analytical chemistry before anyone could read it at all. And the third reason is perhaps the most ironic. We are, biologically, the children of the Great Oxidation Event. Every one of our cells runs on the oxygen that cyanobacteria pumped into the ancient atmosphere. We are so completely built around this event, so thoroughly the product of the world it created, that we have spent most of our history entirely unaware that a different world ever existed, let alone that it was destroyed to make room for the one we inhabit. The largest extinction in the history of life on Earth was not a disaster that happened to someone else. It was the necessary precondition for our own existence. We have simply never been taught to see it that way. If you could travel back to the ancient ocean during the peak of the oxygen crisis and somehow observe what was unfolding beneath the surface, you would witness something for which no modern reference exists. Not a war, because war implies agency and intention. Not a storm, because storms end and leave the world recognizable. What you would see is closer to a slow erasure, the methodical, chemical dissolution of an entire living world, proceeding at the pace of geological time, but with absolute thoroughness. The seafloor was the first place to show the signs. For hundreds of millions of years, the ocean floor had been carpeted in living bacterial mats of extraordinary complexity and color, purple sulfur bacteria in dense, shimmering sheets, orange and red communities of iron-metabolizing organisms, building intricate mineral structures around themselves, green and brown photosynthetic bacteria in the shallower zones, layering themselves into stromatolites, dome-shaped biological formations whose fossilized remains are among the oldest physical evidence of life on Earth. These communities were not simple. They were ecosystems in miniature, with their own internal diversity, their own chemical cycles, their own evolutionary histories stretching back further than any fossil record had been able to document. As the oxygen spread through the water column, these communities began to change. The change was not sudden. It did not sweep through in a wave of visible death. Instead, it worked from the surface downward, from the edges inward, pressing the oxygen-sensitive organisms deeper and deeper as the concentration gradient shifted. The purple bacteria retreated from the zones where oxygen penetrated. The iron-metabolizing communities began to fall apart as the chemistry of their environment shifted away from the reduced iron compounds they required. The delicate balance of the sulfur cycle, a cycle that had been running in continuous, elegant equilibrium for over 1 billion years, began to fracture under the chemical pressure of a molecule it had never been designed to accommodate. The banded iron formations that had been building on the seafloor for hundreds of millions of years, those extraordinary rust-red layers of iron oxide laid down as the ocean's chemical defense against rising oxygen, ceased forming. The iron was gone. The buffer was exhausted, and with it went the last mechanism that had been protecting the ancient biological world from the full force of what the cyanobacteria had unleashed. In the deepest parts of the ocean, where the light never reached and the oxygen penetrated slowly, communities of anaerobic organisms survived in the darkness. These became refugia islands of the Old World, maintained by depth and chemistry, and the sheer persistence of organisms that had been running the same biological programs for 1 billion years. They are still there today. If you drill into the sediment of the deepest ocean trenches, you still find their descendants' ancient anaerobic lineages, running ancient biochemistry, living proof that the Old World was never entirely erased. But on the surface, in the warm shallows, in the mid-water zones where life had been richest, the dying continued. Slow, chemical, absolute. There are catastrophes that arrive from outside asteroids, supervolcanoes, solar flares. They are devastating, but they are at least comprehensible as disasters. Something foreign strikes a system and breaks it. The cause and the effect are separated by a clear boundary. What happened next during the Great Oxidation Event belongs to a different and stranger category. A catastrophe that the system generated entirely from within itself, through the interactions of its own components, producing consequences that bore no intuitive relationship to their cause. Methane had been warming Earth for billions of years. This is not a minor detail. The young sun burned at roughly 70% of its current luminosity when life first appeared on Earth, and by all standard calculations, a planet at Earth's distance from a sun of that brightness should have been completely frozen. A ball of ice with no liquid oceans, no chemistry, no life. The reason Earth was warm instead is almost entirely attributable to methane, produced in staggering quantities, by the methanogen bacteria that dominated the ancient ocean floors. Methane is a greenhouse gas roughly 80 times more potent than carbon dioxide at trapping heat in an atmosphere. The ancient atmosphere was saturated with it, and that saturation was what kept the planet's surface above freezing, despite the dim young sun. Methane and oxygen are chemically incompatible. When oxygen enters an atmosphere rich in methane, it reacts slowly, but relentlessly converting methane into carbon dioxide and water. Carbon dioxide is also a greenhouse gas, but it is dramatically less effective at trapping heat than methane. The conversion of the methane blanket into carbon dioxide represents, in planetary terms, the the removal of a heating system, and its replacement with a much weaker one. As oxygen levels rose during the Great Oxidation Event, this is precisely what happened. The methane that had been keeping Earth warm for billions of years was systematically destroyed, molecule by molecule, by the same oxygen that was simultaneously poisoning the ancient biological world. The greenhouse effect weakened, global temperatures began to fall, and they did not fall gently. The result was the Huronian glaciation, one of the most severe ice ages in Earth's entire history, lasting perhaps 300 million years, during which glaciers extended from the poles all the way to the equator. The entire surface of the ocean may have been covered in ice kilometers thick. Surface temperatures plunged to levels that make modern ice ages look like mild seasonal variations. Scientists call this extreme state snowball Earth, and its evidence, glacial deposits found at ancient equatorial latitudes, distinctive sedimentary signatures of catastrophic freeze-thaw cycles, is preserved in rock formations on multiple continents. Cyanobacteria, in their blind, relentless photosynthetic labor, had not merely poisoned the atmosphere. They had also dismantled the heating system. They had frozen the planet. One organism, two catastrophes, zero intention. The ancient world was being remade, violently and completely, by the simplest kind of life that has ever existed. Not everything died. This is important. This is, in fact, one of the most important facts in the entire history of life on Earth, even though it sounds almost anticlimactic set against the scale of what had been destroyed. The Great Oxidation Event and the Huronian glaciation together represented the most comprehensive transformation of planetary living conditions in biological history. But life, tenacious, chemically inventive, temporally patient in ways that make human endurance seem brief, found places to hide. These places are called refugia, and they were scattered across the ancient planet in forms and locations that would have seemed, to any observer capable of appreciating them, almost impossibly marginal. Deep-sea hydrothermal vents, where heat from Earth's interior created small, oxygen-free oases of chemical energy completely isolated from the oxygenating ocean above. Hypersaline lakes with unique chemical compositions that neutralized oxygen before it could penetrate deeply. Permafrost layers in the proto-continents above the ancient glaciers, where frozen temperatures slowed metabolism to near zero and preserved the cellular machinery of ancient organisms in biological suspended animation. And within the thick microbial mats that had once covered ocean floors, even within these dying communities, an internal chemistry was at work. The deeper layers of the mats, pressed beneath the weight of organism upon organism, existed in oxygen gradients that shielded their innermost communities from the full force of the oxidizing environment above. In these refugia, the old world survived. Not intact, not in the rich, planet-spanning diversity that had characterized the pre-oxygen Earth, but alive, running, persistent. And here is where this story takes a turn that changes everything that follows. In the refugia, in the margins, in the darkest and most chemically extreme corners of a planet that had been turned hostile to almost all existing life, organisms were not simply surviving, they were evolving. The extreme chemical stress of fluctuating oxygen levels, pockets of high oxygen separated by regions of none, boundaries where the two chemical worlds met and mixed, created exactly the kind of selective pressure that drives rapid evolutionary innovation. Organisms that could tolerate a little oxygen, that had stumbled by accident onto some molecular adaptation that gave them an edge in the new chemical environment, had a survival advantage over their neighbors. And survival advantages, multiplied across enough generations, become new capabilities. Something was changing in the refugia. Something was learning, in the only way biology knows how to learn, through the patient, brutal, statistically enormous lottery of mutation and selection, how to not merely endure oxygen, but to use it. The descendants of those ancient refugees still exist today. They live in the oxygen-free sediments of deep lakes, in the airless mud of ocean trenches, in the depths of intestinal tracts, including human ones, running the same ancient anaerobic biochemistry their ancestors ran before the oxygen catastrophe. The old world never fully died. It retreated, it adapted at its margins, and from those margins it was about to produce something the ancient Earth could never have imagined. Evolution does not plan. This cannot be said often enough, because the human mind is a planning mind. It sees structures and assumes architects, sees outcomes and assumes intentions, sees a world shaped by oxygen and assumes that something, somewhere, was working toward that outcome. Nothing was. Evolution operates through the same mechanism it has always operated through: random mutation, differential survival, the statistical accumulation of changes that happen to improve an organism's reproductive success in its current environment. It has no memory of where it is going. It has no awareness of where it has been. And yet, given enough time and enough pressure, it produces results of almost architectural precision. The pressure in the oxygen-tainted margins of the ancient world was unlike anything biology had encountered before. Oxygen doesn't just damage cells at the level of gross chemical disruption it generates as a consequence of its reactions with biological molecules. A second wave of destruction in the form of what chemists call reactive oxygen species. These are molecular fragments with unpaired electrons, superoxide radicals, hydrogen peroxide, hydroxyl radicals that are even more chemically aggressive than oxygen itself. They shred the long molecules of DNA. They puncture lipid membranes. They destroy the three-dimensional structure of proteins that have been folded by evolution into precise functional shapes, shapes that represent billions of years of refinement. For an anaerobic organism with no defense against these molecular vandals, the result was cellular collapse. But in populations large enough and variable enough, and bacterial populations are both, containing more individuals in a single tablespoon of ancient seawater than there are stars in the observable universe, some individuals carried, by pure accident, molecular variants that happened to neutralize these reactive species before they could cause full damage. Perhaps a protein that had evolved for a different purpose could, in its particular shape, bind and neutralize a superoxide radical. Perhaps a metal ion, naturally present in a cell's chemistry, could catalyze the conversion of hydrogen peroxide into water. These were not designed defenses. They were accidental competencies, molecules that happened to have the right shape for a new job. But organisms carrying them survived longer. They reproduced more successfully. Their descendants inherited the same accidental competency and, over millions of generations, refined it through further mutation and selection into something extraordinarily precise: the antioxidant enzyme systems that exist in virtually every living cell on Earth today. Superoxide dismutase, catalase, peroxyredoxins, glutathione. The names are technical, but the function is poetic in its elegance. These molecular bodyguards intercept the reactive products of oxygen chemistry before they can reach the cell's critical machinery, dismantling them into harmless water and oxygen with a speed and precision that modern biochemistry still studies with genuine admiration. Life was not adapting to oxygen as a strategy. It was being forced, by the merciless arithmetic of survival and death, to discover defenses it had never needed before. And once those defenses existed, something else became possible. Something that would change the trajectory of life on Earth forever. Consider, for a moment, what it means to run an entire civilization on candles. Not metaphorically. Actually imagine it: every industrial process, every form of transportation, every tool and machine, powered by the gentle, flickering combustion of wax. You could build things with candles. You could heat spaces, light rooms, drive certain very simple mechanisms with enough of them, arranged cleverly enough. But there are things you simply could not do. You could not build a skyscraper, because the structural calculations require precision manufacturing tools that need more power than candles can provide. You could not fly, because flight requires energy densities that candlelight cannot approach. You could not think particularly complex thoughts at an organizational level, because complex thought at scale requires communication infrastructure that demands far more than flame. Now imagine someone hands you a jet engine. This is, with only slight exaggeration, what the evolution of aerobic respiration meant for the biological world. Before oxygen, all life on Earth ran on anaerobic metabolism, a set of biochemical processes for extracting energy from organic molecules that are, by the standards of what came later, extraordinarily inefficient. The most common form of anaerobic energy extraction, fermentation, yields a net gain of approximately 2 ATP molecules per glucose molecule. ATP adenosine triphosphate is the universal energy currency of living cells, the molecule that powers everything from muscle contraction to protein synthesis to the electrical signals of neurons. 2 molecules per glucose, that is the energy budget of the anaerobic world. That is all the power the candles provided. Aerobic respiration, using oxygen as the final electron acceptor in a carefully organized electron transport chain, yields approximately 36 to 38 ATP molecules from the same glucose molecule, not twice as much. Not 5 times as much, 18 to 19 times as much, from identical raw material. The implications of this energy revolution cascade through every level of biological possibility. With 18 times more energy available, organisms could afford to grow larger, dramatically larger, since the metabolic cost of maintaining a bigger body scales predictably with mass. They could afford to move faster, since movement is energetically expensive, and the anaerobic budget barely covered the cost of the microscopic motility that bacteria had achieved. They could afford to build more complex cellular machinery, since every additional protein, every additional membrane structure, every additional biochemical pathway has an energy cost that must be met continuously just to maintain it. Most significantly, they could afford complexity itself. Not just the complexity of a single large cell, but the staggering complexity of multiple cells working in coordination, cells that had specialized for different functions, cells that had divided the labor of survival between different tissue types, cells that formed structures whose capabilities vastly exceeded anything a single cell could achieve alone. And here is where the story becomes deeply personal, where the events of 2.5 billion years ago reach forward through geological time and touch you, specifically, in this specific moment. The mitochondria in every cell of your body, those small, membrane-wrapped organelles that perform the aerobic respiration keeping you alive, are not originally human structures. They are not inventions of the eukaryotic cell that contains them. They are the direct descendants of ancient bacteria that independently evolved aerobic respiration and were then captured, integrated, and preserved inside a host cell in one of the most consequential accidents in the history of life. The jet engine inside you is an ancient, repurposed bacterium. It still carries its own DNA. It still divides on its own schedule. It is still, in a very precise biological sense, a separate organism living inside you. Somewhere in the ancient ocean, in the turbulent chemical borderlands between the old anaerobic world and the rising oxygen tide, a meeting took place that changed the course of life on this planet more fundamentally than any other single event in biological history. An archaean cell, a single-celled organism belonging to one of Earth's 2 most ancient domains of life, engulfed an aerobic bacterium. This was not unusual in itself. Engulfment was a common predatory strategy in the microbial world, a way of extracting nutrients from other organisms by surrounding them with membrane and digesting them from the inside out. The archaean had almost certainly done this before, many times, with many different prey organisms. But this time, something failed, or something succeeded, depending entirely on how you choose to measure the outcome. The bacterium was not digested. Perhaps it had a chemical defense that inhibited the host cell's digestive enzymes. Perhaps the particular stress conditions of the oxygen transitional environment had disrupted the host's normal digestive mechanisms at a critical moment. Whatever the reason, the aerobic bacterium survived inside its captor, and instead of becoming a meal, it became a partner. The bacterium was already producing energy through aerobic respiration, using the oxygen that was flooding the ancient environment and generating ATP with that extraordinary 1/8-fold efficiency. Trapped inside the host cell, it continued doing exactly what it had always done. And the host cell, which had been struggling to compete in an oxygenating environment with its ancient inefficient anaerobic metabolism, found itself suddenly and accidentally in possession of an internal power generator of previously unimaginable productivity. Over millions of generations, the partnership deepened into something neither party could undo. The internal bacterium lost the genes for independent survival one by one, transferring many to the host cell's nucleus, where they could be regulated by the host's own genomic machinery. The host cell became progressively dependent on its internal power source, restructuring its own biology around the energy supply it now relied on absolutely. The boundary between the two organisms blurred, generation by generation, until what had begun as predator and prey became something entirely new, a single, integrated cell of unprecedented biological complexity. That cell became the ancestor of every eukaryote on Earth, every animal, every plant, every fungus, every organism complex enough to be visible to the naked eye. Every creature that has ever swum, walked, flown, burrowed, or bloomed on this planet carries the descendant of that ancient captured bacterium, now called the mitochondrion, running quietly inside every cell, powering every heartbeat, every breath, every thought. The accident that started everything is still running, right now, inside you. Once the energy barrier was broken, evolution began building things it had never been able to afford before. For over 3 billion years, life on Earth had been single-celled. This was not a failure of imagination. Evolution has no imagination. It was a failure of resources. A single cell running on anaerobic metabolism simply cannot accumulate and maintain the molecular machinery required to coordinate with other cells in a stable, functional way. The energetic cost of cellular communication, of building specialized structures, of maintaining the biochemical channels through which multiple cells could behave as a unified organism rather than a collection of independent competitors. These costs exceeded what the candle-powered anaerobic budget could sustain. With aerobic respiration and the mitochondrial energy revolution, that budget was multiplied by nearly 20, and with it, an entirely new category of biological possibility opened. Multicellularity did not arrive in one great creative leap. It arrived in experiments, small, tentative, sometimes unsuccessful experiments that were repeated independently across multiple biological lineages over hundreds of millions of years. Certain cyanobacteria had already begun forming simple filaments, chains of connected cells that remained attached after division, creating elongated structures with primitive division of labor between cells at different positions in the chain. Early colonial algae discovered that clusters of cells could feed more efficiently than isolated individuals. Simple aggregates of similar cells found that their collective chemistry created environmental conditions no individual cell could achieve alone. Each experiment solved a specific problem. Some solved the problem of size. A larger body, even if built from many cells rather than one larger one, could access food sources and resist predation in ways unavailable to a microscopic individual. Some solved the problem of specialization. If different cells in a cluster could take on different functions, the overall organism could perform multiple tasks simultaneously that no single cell could manage alone. Some solved the problem of persistence. Multicellular organisms could survive the death of individual cells in ways that single-celled organisms, where cell death equaled organismal death, could not. The evolutionary advantages compounded. More energy enabled larger bodies. Larger bodies created selective pressure for better organization. Better organization enabled more sophisticated behavior. More sophisticated behavior created new ecological niches. New niches created new evolutionary pressures. The wheel, once turning, accelerated. Something else was building beneath all of this. Biological innovation. Something invisible, atmospheric, patient. The same cyanobacteria that had triggered the catastrophe had never stopped photosynthesizing. Oxygen had been accumulating in the atmosphere for hundreds of millions of years. And as the oxygen concentration rose, it was doing something in the upper atmosphere that no organism had planned for, no evolutionary process had aimed toward, but that every land-based life form in Earth's entire future would depend on absolutely. It was building a shield. 541 million years ago, something happened in the world's oceans that scientists have been arguing about, marveling at, and attempting to explain ever since its discovery in the fossil record. In a window of geological time that seems almost impossibly brief, perhaps 20 to 25 million years, which is the blink of an eye measured against the 4.5 billion year duration of the planet, the fossil record transforms from sparse and simple to rich and bewildering. Before this moment, the visible fossil record is dominated by soft-bodied organisms of mysterious and often unclassifiable form. The strange quilted creatures of the Ediacaran period, flat and featureless, seemingly without mouths or organs, more like biological abstractions than recognizable animals. After this moment, the oceans are full of creatures with eyes, with legs, with shells, with claws, with complex sensory systems and sophisticated body plans. Trilobites with compound eyes of extraordinary optical precision. Anomalocarids, ancient apex predators, the largest animals alive, built like armored submarines with grasping appendages and circular, multi-part mouths. Early filter feeders whose descendants would become the sea lilies, still decorating modern oceans. And, crucially, small, soft-bodied creatures, with a stiff rod running along their backs, the notochord that would eventually evolve through hundreds of millions of years of further development into the vertebral column that structures every fish, every amphibian, every reptile, every bird, and every mammal that has ever existed. Every major animal body plan represented in the living world today has its origin in this extraordinary creative burst. The Cambrian explosion is biology's most spectacular flowering, and it happened with remarkable directness because of oxygen. The connection operates at several levels simultaneously. The most fundamental is energetic. Complex, active animals with sophisticated nervous systems, muscular locomotion, and hard mineralized body parts require metabolic energy budgets that simply could not have been supported at the lower oxygen concentrations of the preceding eons. As atmospheric oxygen rose and dissolved oxygen in the oceans increased correspondingly, the energetic threshold for increasingly complex body plans was successively crossed. Larger nervous systems became metabolically viable. More sophisticated sensory organs became affordable. Faster, more powerful muscular systems became sustainable. But oxygen did something else too. It drove an arms race. The first predators with hard parts and active hunting strategies created, for the first time in Earth's history, a powerful and immediate selective pressure for prey organisms to develop defenses. Shells, spines, burrowing behaviors, camouflage, speed. Predators responded with more sophisticated hunting tools. Prey responded with better defenses. The cycle accelerated, and with it, biological diversity exploded in what evolution theorists describe as a positive feedback loop of ecological complexity. An ocean without predators is an ocean without much evolutionary urgency. An ocean with predators is an ocean in a constant species-driving arms race that pushes biological innovation faster than any other known evolutionary force. The Cambrian explosion was not a mystery. It was an answer. The question it was answering had been accumulating in the atmosphere, molecule by molecule, for nearly 2 billion years. And the world it created, the world of active, complex, diverse animal life, is the world that every subsequent chapter of life on Earth was written in. For most of Earth's history, the land was barren. Not desert barren, the way certain regions of the modern world are barren. Completely barren rock and dust, and raw mineral surfaces, with no soil, no roots, no spores, no crusts of lichen, no microbial films on stone. Just geology, unmediated by biology, baking under a sun whose ultraviolet radiation reached the surface completely unfiltered and sterilized, every surface it touched with the indifferent thoroughness of a planetary autoclave. The reason for this absolute sterility was simple. Earth's atmosphere, in its pre-oxygen state, had contained no ozone. Ozone, the molecule, consisting of 3 oxygen atoms bound together, rather than the usual 2 forms in the upper atmosphere, when ordinary oxygen molecules are struck by ultraviolet radiation from the sun and split apart, creating free oxygen atoms that recombine with other oxygen molecules in the 3-atom configuration. No atmospheric oxygen means no ozone. No ozone means no protection from solar ultraviolet radiation. And without protection from ultraviolet radiation, any organism that ventures onto a land surface is exposed to a sterilizing energy flux that tears apart DNA and destroys proteins with lethal efficiency. But oxygen had been accumulating. For hundreds of millions of years after the Great Oxidation Event, atmospheric oxygen concentrations had been rising slowly, episodically, with periods of plateau and renewed increase, and with every increment of oxygen, the ozone layer above grew fractionally thicker, fractionally more protective. The first organisms to test the shielded land were among the most resilient on Earth, cyanobacterial crusts, capable of surviving desiccation, temperature extremes, and residual ultraviolet doses that would kill most organisms. They spread slowly across ancient rock surfaces, breaking them down chemically, contributing organic material that, over millions of years, became the first primitive soil. Lichens followed those extraordinary partnerships between fungi and photosynthetic organisms, and then primitive plant-like organisms, and then the first true vascular plants, and then forests. And then, following the food that the forests represented, animals. Arthropods, first ancient relatives of scorpions and millipedes, armored and desiccation-resistant, moving cautiously onto a land still only partially hospitable. Then, much later, the lobe-finned fish, whose descendants developed weight-bearing limbs and learned to breathe air. Then, amphibians. Then reptiles. Then, in the fullness of geological time, mammals. Every one of these transitions was made possible by the oxygen shield overhead. The cyanobacteria that had poisoned the ancient oceans had also, over the following 2 billion years, built the atmospheric armor that made every single land-based life form in Earth's history possible. The destroyers had become, without knowing it, the architects of everything. There are places on Earth where you can stand on rock that is older than almost anything else on the planet's surface, and if you know what you are looking at, you can read a story in the stone that no written language could have preserved. In the ancient cratons of Western Australia, in the shield rocks of northern Canada, in the Precambrian formations of South Africa, there are cliff faces and canyon walls where the rock is striped layer after alternating layer of rust-red iron oxide and dark grey silica, stacked with a regularity that looks almost deliberate, almost architectural, as though some enormous and patient hand had been alternating the colours of the stone. These are banded iron formations, and they are the world's oldest crime scene. Each layer is a chapter. The rust-red bands are periods when free oxygen was available in the ancient ocean. Iron dissolved in the seawater reacted with the oxygen produced by cyanobacteria, forming iron oxide that precipitated out of solution and drifted to the sea floor in a slow, rust-coloured snow. The dark bands are periods when oxygen levels dropped, iron remained in its dissolved form, and silica-rich sediment settled in its place. The alternation of the two types of layers traces, in stone, the fluctuating chemistry of an ocean caught between the oxygen-producing cyanobacteria above and the iron-rich geological chemistry of the seafloor below, a record of a battle between biological production and chemical absorption that lasted for hundreds of millions of years. The global extent of these formations is staggering. BIFs appear on every continent, in rock formations that have been dated to the same geological window, all telling the same chemical story in the same chemical language. The convergence is not coincidental, it is the signature of a planet-wide chemical transformation, a simultaneous shift in ocean chemistry that left the same mineralogical record in every ocean basin simultaneously. And then, as abruptly as they began, the BIFs stop forming. Around 1.8 billion years ago, they largely disappear from the geological record, because the dissolved iron that had been their raw material was exhausted, consumed by the very oxygen whose accumulation they had been recording, and the ocean's chemistry had shifted permanently into a new state. The formations we have are all that remains of the ancient ocean's long chemical struggle, and they are not merely scientifically invaluable, they are economically critical. The iron ore deposits of the Pilbara in Australia, the Mesabi Range in North America, the vast formations of Brazil and South Africa, these are the world's primary sources of iron for industrial steel production. Every steel beam in every building, every car chassis, every ship hull, built from the fossilized chemistry of the world's greatest biological catastrophe. The crime scene turned out to be the raw material of civilization. Rock layers can tell you what happened, but determining precisely when it happened and proving that the atmospheric chemistry changed in the way the banded iron formations suggest required a different kind of evidence, one that came not from the minerals themselves but from the isotopes hidden within them. Sulfur has 4 stable isotopes, atoms of sulfur with slightly different masses, and in most chemical and biological processes, these isotopes are fractionated in predictable, mass-dependent ways. Heavy isotopes react fractionally more slowly than light ones, and this difference is regular, consistent, and well understood. Under normal circumstances, measuring the ratios of sulfur isotopes in ancient sediments tells you about the chemistry and biology of the environment in which they were deposited. But under one very specific set of circumstances, something different happens. In an atmosphere with no ozone layer, an atmosphere with no significant oxygen, ultraviolet radiation from the sun reaches the lower atmosphere and drives photochemical reactions in volcanic sulfur gases that produce a different kind of isotope fractionation, a fractionation that does not follow the mass-dependent rules, a fractionation that is, in scientific terms, mass-independent, a signature that can be produced only by ultraviolet photochemistry in an ozone-free atmosphere, and that disappears completely the moment an ozone layer forms and blocks the ultraviolet radiation that drives it. This mass-independent sulfur isotope signature is present in ancient sedimentary rocks from before 2.4 billion years ago. It is absent from rocks deposited after 2.4 billion years ago. The boundary is sharp, clear, and global. The same transition appears in ancient rock formations from multiple continents, all converging on the same geological moment. Scientists call this the Great Oxidation Event's clearest chemical timestamp, a switch in the isotopic record that marks, with remarkable precision, the moment when atmospheric oxygen reached concentrations sufficient to form a persistent ozone layer and permanently shut down the ultraviolet photochemistry that had been producing the distinctive signature for billions of years. Combined with the BIF record, with the disappearance of certain oxygen-sensitive minerals from ancient river delta deposits, with the appearance of oxidized red beds in continental sediments, and with isotopic signatures in ancient carbon and nitrogen records, this sulfur signal locks the Great Oxidation Event to a specific and well-constrained window of geological time. Multiple independent lines of evidence, each developed through different analytical techniques, each preserved in different types of ancient rock from different continents, all pointing to the same moment. Science does not often achieve this kind of convergence. When it does, what it is describing is not a theory, it is the past. The rocks remember, the isotopes remember, and, in a way that is perhaps the most remarkable of all, the living organisms of today still carry the memory of the Great Oxidation Event inside their genomes. A biological archive of the catastrophe, written not in stone, but in the sequence of DNA that has been copying itself, with small errors and large consequences, for over 2.5 billion years. The molecular clock is one of evolutionary biology's most powerful tools. It is based on a straightforward observation. Neutral mutations, changes to DNA sequences that have no significant effect on an organism's function, accumulate at roughly predictable rates over time. If you compare the genomes of two organisms and count the differences in their neutral sequences, you can estimate how long ago their lineages diverged from a common ancestor. The longer the separation, the more differences have accumulated. The clock is imprecise, mutation rates vary between organisms and over time, and the calibration requires fossil evidence to anchor specific dates, but applied carefully and cross-referenced against multiple genomic regions, it provides genuinely useful estimates of evolutionary divergence times. When evolutionary biologists applied the molecular clock to the major biological transitions associated with the Great Oxidation Event, the results were extraordinary. The origin of the key antioxidant enzyme families, the superoxide dismutases, the catalases, the peroxidoxins dates to approximately the same period as the geological evidence for the oxygen transition, providing direct genomic confirmation that these defensive innovations appeared in response to the rising oxygen challenge. The divergence of major aerobic metabolic lineages from their anaerobic predecessors clusters around the same deep time frame. The endosymbiotic event that created the mitochondrion traces, through both the genomic similarity between mitochondrial DNA and certain bacterial lineages, and the molecular clock evidence in mitochondrial and nuclear genomes, to a period consistent with the early aftermath of the Great Oxidation Event. But perhaps the most remarkable genomic evidence comes from the organisms that did not adapt the surviving anaerobes that retreated into the refugia and have been running their ancient biochemistry ever since. Their genomes carry molecular machinery that predates the Great Oxidation Event, enzyme systems, and metabolic pathways that were already ancient when oxygen first appeared, preserved in continuous operation for over 2.5 billion years. They are biological time capsules, living archives of the pre-oxygen world, and they still exist in the deep sediments of oxygen-free environments on every continent. 3 completely independent scientific disciplines, geology, isotope geochemistry, and molecular genetics, have each read the record of the Great Oxidation Event from entirely different archives using entirely different methods. They agree, down to the same geological moment, the same biological transitions, the same chemical thresholds. The past, it turns out, never stops leaving evidence. Right now, in orbit around Earth, and aimed at the atmospheres of planets orbiting distant stars, telescopes are searching for a specific chemical signal, a combination of molecules in an alien atmosphere whose presence would be considered by many scientists as the strongest possible evidence of life beyond our solar system. The signal they are looking for involves oxygen. More specifically, it involves the simultaneous presence of oxygen and methane in the same atmosphere, a chemically unstable combination that destroys itself in a matter of thousands of years and can only be continuously replenished by active biological processes. This is, with extraordinary historical irony, the same chemical signature that Earth itself produced during and after the Great Oxidation Event. The logic of the biosignature is impeccable. Oxygen is, as we have seen, a reactive gas that does not persist in an atmosphere without continuous replenishment. On Earth, that replenishment comes from photosynthesis, the same cyanobacterial process that originally oxygenated the ancient atmosphere, still running today in the cells of every plant and algae on the planet. Remove the photosynthesis, and Earth's atmospheric oxygen would be consumed by surface chemistry within a few million years, a geological eye blink. The presence of oxygen in substantial concentrations in any planetary atmosphere is therefore a very strong indicator that something biological is producing it continuously. Methane persists even less readily in an oxygenated atmosphere. It reacts with oxygen and disappears on timescales of decades. Its presence alongside oxygen in a stable planetary atmosphere requires an equally continuous biological source, the same methanogen bacteria whose ancient predecessors had maintained Earth's methane blanket before the oxygen catastrophe depleted it. For astronomers scanning the atmospheric spectra of exoplanets, analyzing the characteristic wavelengths of light absorbed by different molecules as starlight passes through a planet's atmosphere during a transit, the LIX combination of oxygen and methane in the spectrum of an Earth-sized world in its star's habitable zone would be, as one prominent astrobiologist described it, the closest thing to a signed confession of biological activity that the universe can provide. And this is the extraordinary legacy of the cyanobacteria and the catastrophe they caused. The same chemical transformation that killed the ancient world, that froze the planet, that drove the greatest mass extinction in Earth's history, produced the atmospheric signature that astronomers now regard as the universal marker of biological life. The Great Oxidation Event did not just change Earth, it defined what a biologically inhabited planet looks like from space. It wrote the template that the entire search for extraterrestrial life is now built around. We are not looking for alien life in the abstract. We are looking for other planets that have been transformed by their own biology, the way ours was looking for other great oxidation events, written in the spectra of distant atmospheres, waiting to be read. Intelligence is expensive. This is not a philosophical observation, it is a metabolic one, and it is one of the most important and underappreciated facts about the nature of complex cognition. The human brain is the most energetically costly structure in the known universe relative to its size. It represents approximately 2% of the body's total mass and consumes somewhere between 20 and 25% of its total resting metabolic energy. This extraordinary proportion means that maintaining a functioning human brain, not thinking hard, not solving problems, just maintaining the baseline electrical activity that keeps a brain alive and operational, requires a continuous, high-intensity supply of aerobic metabolic energy that the brain's neurons are extraordinarily sensitive to. Cut oxygen supply to the brain for 4 minutes, and consciousness begins to fail. Cut it for 10, and the damage is permanent. Cut it for 20, and the brain is dead. This is not a design flaw. It is the inevitable consequence of what a brain is, a structure of almost unimaginable electrochemical complexity, maintaining billions of simultaneous connections, running continuous computational processes at speeds that make human-designed computers look pedestrian, all powered by the aerobic metabolism of billions of individual cells working in continuous coordination. In an anaerobic world, this structure is simply impossible. Not difficult, not rare. Impossible because the energy budget of anaerobic metabolism cannot sustain the metabolic demands of complex neural tissue at the required scale. The math is unambiguous. 2 ATP molecules per glucose molecule cannot power a brain. The candle cannot run the jet engine. Intelligence, as we know it, as any neurological evidence suggests it could exist, is therefore a direct and specific consequence of aerobic respiration. It is a consequence of the energy revolution that was triggered by the Great Oxidation Event. It is a consequence of the cyanobacterial oxygen catastrophe. The implications for the search for intelligence elsewhere in the universe are profound and somewhat sobering. If intelligence requires high-energy aerobic metabolism, and aerobic metabolism requires atmospheric oxygen, and atmospheric oxygen requires photosynthetic life of a specific and sophisticated type, and the development of photosynthetic life requires a specific sequence of geological and chemical conditions to develop and persist, then the chain of prerequisites for intelligence is long, specific, and potentially rare. Every link in that chain must be present and functional for the final outcome to appear. Remove any link, prevent the development of oxygenic photosynthesis, prevent the ocean chemistry from crossing the oxidation threshold, prevent the endosymbiotic merger that created the mitochondrion, and intelligence as we understand it never arrives. We may be rare not because the universe is unfriendly to life, but because the specific path from life to intelligence requires a chain of accidents whose simultaneous occurrence may be extraordinarily unlikely. There is a pattern that runs through Earth's geological history, visible only at the scale of deep time but unmistakable once you know to look for it. The pattern is this: biological activity changes atmospheric chemistry. Changed atmospheric chemistry disrupts the conditions that existing biological communities depend on. Disrupted conditions trigger cascading consequences that no individual organism anticipated or intended, and no single organism could have prevented. And the world that emerges from the cascade is transformed, not destroyed, because life has proved extraordinarily difficult to extinguish entirely, but transformed into something that the organisms living before the transformation could not have recognized or survived. This pattern has run once at maximum scale in the Great Oxidation Event. It has run in smaller iterations throughout geological history, and it raises a question that the geological record makes genuinely difficult to avoid. The current carbon dioxide concentration in Earth's atmosphere is rising at a rate that has no geological parallel in the ice core and sedimentary record for at least the last several million years. The source is biological, the combustion of organic carbon by one specific species, just as the source of the ancient oxygen rise was biological, the photosynthetic activity of one specific group of organisms. The speed of the atmospheric change is, in both cases, fast, by geological standards, faster than the natural buffering systems of the planet can fully absorb. And in both cases, the organisms driving the change are not doing so with any awareness of the planetary consequences. The cyanobacteria had no choice. They followed their chemistry. The Great Oxidation Event ultimately produced a world of staggering biological richness, more complex, more diverse, more energetically vibrant than anything the ancient anaerobic world had achieved. But the transition took hundreds of millions of years and involved the largest mass extinction in Earth's history. The organisms living through the transition did not experience it as an exciting evolutionary opportunity. They experienced it as the end of everything they had ever known. The geological record is not a morality tale. It does not offer conclusions or prescriptions. What it offers is perspective, the perspective of a planet that has been through radical atmospheric transformation before, that has records of what such transformations produce, and that has no mechanism of its own for caring about the outcome. The organisms of the ancient anaerobic world could not read the banded iron formations building on their seafloor. They could not see the oxygen accumulating in the water above them. They had no way to know what was coming. We are the only organisms in Earth's history that have ever had the ability to read the record. The question of what we do with that ability belongs entirely to us. They are everywhere, and almost no one thinks about them. Right now, in the surface waters of every ocean on Earth, in freshwater lakes and rivers and puddles, in the thin biological crusts of desert soils, in the ice of polar regions and the scalding water of geothermal springs, cyanobacteria are photosynthesizing. They are splitting water molecules, absorbing carbon dioxide, releasing oxygen, the same fundamental chemistry they have been performing for over 3.5 billion years, unchanged in its essential mechanism by the entire span of biological time. They produce somewhere between 20 and 30% of all atmospheric oxygen replenishment on Earth. They fix atmospheric nitrogen in forms accessible to other organisms, contributing enormously to the global nutrient cycles that underpin all ecosystems. They are, in the most literal possible sense, still running the world, and their influence extends far beyond their direct activity. Inside every plant cell and every algal cell on Earth, there are structures called chloroplasts, the green organelles that perform photosynthesis in plant tissue, that give leaves their color and make the energy conversion that feeds essentially every food chain on land. Chloroplasts are not original inventions of plant cells. They are captured cyanobacteria, the product of a second endosymbiotic merger, similar to the one that created mitochondria, in which an ancestral eukaryotic cell engulfed a cyanobacterium and, instead of digesting it, incorporated it permanently as an internal photosynthetic factory. This means that every forest that has ever grown on this planet, every meadow, every algal bloom, every kelp forest, every microscopic phytoplankton in the open ocean, all of it photosynthesizes using biochemical machinery that is ultimately a modified form of cyanobacterial biology. The organisms that accidentally created the oxygen catastrophe are also the direct ancestors of every photosynthetic process that has ever occurred on Earth. Their resilience matches their influence. Cyanobacteria survive conditions that would destroy virtually any other group of organisms. The ultraviolet deserts of high-altitude environments, the complete desiccation of desert crusts, the perpetual darkness of sub-ice environments, the extremes of pH and salinity that make certain environments chemically hostile to almost everything else. They have been tested by every catastrophe Earth has experienced in 3.5 billion years of biological history, and have survived all of them. The organisms that poisoned the ancient world, froze the planet, and triggered the largest mass extinction in Earth's history have been continuously photosynthesizing for longer than multicellular life has existed. They have outlasted everything they destroyed, everything that replaced what they destroyed, and every catastrophe that has occurred in the billions of years since. If there is a most successful organism in the history of this planet, this is it. The same chemistry that changed the world once is now being deliberately recruited to change it again, this time not by accident, but by design. In research laboratories on multiple continents, biotechnologists are working with cyanobacteria as platforms for producing things that our current industrial chemistry generates through expensive, energy-intensive, and polluting processes. Cyanobacteria, given sunlight, water, and carbon dioxide, can be engineered to produce ethanol and other biofuels directly from photosynthesis, essentially bypassing the long geological process of fossil fuel formation and producing liquid energy in a matter of days rather than millions of years. Strains have been developed that produce the chemical precursors for biodegradable plastics, for industrial solvents, for pharmaceutical compounds, all from solar energy and atmospheric carbon dioxide, with oxygen as the only other output. The potential scale of this application is difficult to overstate. Every industrial chemical currently produced from fossil hydrocarbons could theoretically be produced photosynthetically using cyanobacterial metabolism as the reaction engine. The same organisms that inadvertently oxygenated the atmosphere might, with sufficient engineering ingenuity, become the biological foundation of an industrial system that runs on sunlight instead of buried carbon. In astrobiology, proposals for the biological terraforming of Mars have identified cyanobacteria as the most plausible candidate for a first-wave terraforming organism, an organism robust enough to survive Mars's extreme radiation, environment in shielded subsurface conditions, capable of beginning the slow process of atmospheric modification that any future Mars colonization would require, and already possessing the biochemical toolkit for performing exactly the kind of atmospheric transformation that would be needed. It would be, in every meaningful sense, a deliberate reenactment of the Great Oxidation Event, the controlled, intentional use of cyanobacterial oxygen production to transform the atmosphere of a planet from uninhabitable to potentially livable. The most fundamental cyanobacterial chemistry is also the subject of intensive research for clean energy applications. The oxygen evolving complex, the protein cluster at the heart of photosystem 2 that performs the water-splitting reaction, is one of the most studied molecular machines in biochemistry, not because of its historical significance, but because of what it does. It splits water into hydrogen and oxygen using only sunlight as its energy source. Hydrogen is a clean fuel. Sunlight is free. Building an artificial version of this molecular machine, artificial photosynthesis, has been identified as one of the most promising potential pathways to sustainable energy production. The ancient chemistry that accidentally transformed a planet is still, 2.5 billion years later, more elegant and more efficient than anything human engineering has managed to build. Here is something the entire story of the Great Oxidation Event asks you to sit with. The cyanobacteria had no intention. This is not a simplification or a poetic shorthand. It is a precise biological statement. Intention requires a nervous system that does not exist at the single-cell level, a capacity for modeling future states and choosing between them that is built on neural complexity that these organisms never possessed and never needed. They split water molecules because their biochemistry instructed them to split water molecules. They released oxygen because that is what the water-splitting reaction produces. They multiplied because that is what successful organisms do. They spread across every sunlit surface on the planet because that is where the sunlight was. And in doing so, they committed the largest mass extinction in Earth's history, froze the planet for 300 million years, and created the conditions for every complex life form that has ever existed, including the one reading these words. We carry a deep intuitive assumption that consequences of this magnitude must have causes of proportional weight, that something this transformative must have been driven by something of comparable intentionality, comparable power, comparable awareness of what it was doing. The Great Oxidation Event dismantles this assumption so completely that it takes a moment to fully absorb the demolition. The most consequential biological event in Earth's history was caused by organisms with no brain, no plan, no awareness, and no capacity for anything that could be called decision-making. They simply followed their chemistry. What does this mean for how we understand our own existence? If you trace the chain far enough, and we have traced it in these pages, from the first cyanobacterium splitting a water molecule in a warm, ancient ocean, through catastrophe and ice, and evolutionary invention, and the flowering of complex life, you arrive at something both humbling and extraordinary. The consciousness that you are using right now to process these words, the awareness that allows you to contemplate what happened 2.5 billion years ago, the mind that finds this story strange and beautiful and unsettling, all of it descended through an unbroken chain of chemical accidents, from an organism that had none of it. The universe, through the blind mechanics of chemistry and time, produced something capable of looking back at its own origin. Whether that means anything is a question the cyanobacteria cannot help you answer. Science has a way of presenting its conclusions that can make them seem more settled than they are textbooks. In particular, tend to crystallize the current best understanding into declarative sentences that carry the full authority of established fact, when what they are actually describing is the leading edge of ongoing investigation. Science has a way of presenting its conclusions that can make them seem more settled than they are textbooks. In particular, tend to crystallize the current best understanding into declarative sentences that carry the full authority of established fact, when what they are actually describing is the leading edge of ongoing investigation, a temporary consensus subject to revision the moment new evidence emerges from a rock face in Australia or an isotope analysis in a Canadian laboratory. The timing and nature of the Great Oxidation Event is, in this sense, very much a living question. The classic model places the transition at approximately 2.4 billion years ago. A relatively sharp rise in atmospheric oxygen from essentially zero to detectable concentrations, recorded in the sulfur isotope signature and the mineral transitions of ancient rock. This model has served well and is supported by a substantial body of geological evidence that has been accumulating since the mid-20th century, but it is not unchallenged. Recent analyses of ancient sedimentary sequences, particularly from the Archean-aged rocks of South Africa's Kapval Craton and the ancient formations of Western Australia, have revealed something unexpected. Scattered through rock deposits predating the classic 2.4 billion year boundary, there are subtle but real chemical signatures suggesting brief, transient spikes in oxygen concentration moments when atmospheric oxygen may have risen to measurable levels before falling back towards zero. Scientists call these oxygen whiffs, and their existence raises a question that cuts to the heart of the Great Oxidation Event's narrative. If cyanobacteria were producing oxygen, and if that oxygen was occasionally reaching the atmosphere in detectable quantities, hundreds of millions of years before the classic Great Oxidation Event boundary, why did it take so long for the permanent transition to occur? What was suppressing the oxygen rise for hundreds of millions of years? What changed at approximately 2.4 billion years ago that allowed the permanent transition to happen then rather than earlier or later? Several hypotheses compete. Some researchers argue that volcanic activity during the early Archean was releasing reducing gases, hydrogen, sulfur dioxide, methane, at rates sufficient to mop up the oxygen before it could accumulate. Others suggest that the continental configuration of the ancient Earth may have been critical, with the arrangement and weathering of early land masses controlling the flux of nutrients and chemical buffers in ways that regulated the oxygen balance. Still others point to changes in the composition of the deep ocean as potentially rate-limiting. The honest answer is that the mechanism is not yet fully understood. The Great Oxidation Event happened. When and why it happened when it did, rather than earlier or later, remains one of the most actively debated questions in all of Earth science. The history of science about the deep past is a history of increasingly precise instruments pointed at increasingly ancient material, producing increasingly surprising results. Every generation of researchers has looked at the same rocks that previous generations examined, and found things in them that were simply invisible to earlier techniques. Chemical signals too subtle for older analytical methods, isotopic ratios requiring precision instruments that did not exist a decade ago, biological signatures requiring genomic tools that were science fiction within living memory. The Great Oxidation Event story has been rewritten significantly in the last 2 decades alone, and the rewriting is ongoing. Molecular clock studies evolutionary biologists using the accumulated mutations in modern genomes to estimate ancient divergence times have pushed the origin of cyanobacteria significantly further back than the geological evidence for the permanent oxygen rise. The best current genomic estimates suggest that cyanobacteria may have originated somewhere between 2.7 and 3 billion years ago, potentially hundreds of millions of years before the classic Great Oxidation Event boundary. If true, this deepens the central puzzle. Why did organisms producing oxygen through photosynthesis for hundreds of millions of years leave no permanent atmospheric signature until 2.4 billion years ago? Meanwhile, discoveries of exceptionally preserved microfossils in ancient Australian and South African cherts, silica-rich rocks that can preserve cellular structures with extraordinary fidelity, have begun to push back the evidence for complex microbial communities into deeper and deeper time, revealing biological structures of surprising morphological complexity in rocks approaching 3.5 billion years old. Recent isotopic analysis of nitrogen compounds in ancient sediments has opened an entirely new window into the nutrient chemistry of the early ocean, suggesting that the availability of biologically accessible nitrogen, a critical nutrient for all life, may have been a significant factor in regulating the timing of the oxygen transition by controlling how much cyanobacterial growth the ancient ocean could support. And, in an entirely unexpected direction, genomic analyses of modern anaerobic organisms have revealed that some of them carry partial aerobic metabolic machinery, enzyme systems, that can process small amounts of oxygen, even in organisms that are fundamentally anaerobic. This suggests that the biochemical toolkit for handling oxygen may have been evolving in the microbial world long before the atmosphere became permanently oxygenated, implying a far longer and more complex biological preparation for the oxygen world than the geological record alone had suggested. Every new technique points into the same ancient darkness and comes back with something that makes the story simultaneously more complicated and more interesting. The Great Oxidation Event is not a closed chapter of geological history. It is an active research frontier, with major discoveries still being made. Looking backward through 2.5 billion years of Earth history reveals something that looking only at the present obscures, that the conditions supporting complex life on this planet are not permanent features of a stable, the world but temporary arrangements in a continuously changing system, and that the direction of that change, on the longest timescales, points toward an end as certain as the beginning. The Sun is not constant. It is a middle-aged star on the main sequence of stellar evolution, and like all such stars, it is slowly and steadily increasing its luminosity as hydrogen fusion in its core converts mass to energy over geological time. Since Earth's formation, solar luminosity has increased by approximately 30%, the same increase that, paradoxically, helped trigger the Great Oxidation Event by intensifying the surface energy available for photosynthesis. Over the next billion to 2 billion years, solar luminosity will continue to rise. The consequences for Earth are predictable and sobering. As the sun brightens, surface temperatures on Earth will rise, despite all natural feedback mechanisms. At some point, estimated by current models at roughly 1 billion years in the future, the increased temperature will accelerate the weathering of silicate rocks to the point where carbon dioxide is drawn down from the atmosphere faster than volcanic activity can replenish it. Carbon dioxide concentrations will fall below the minimum threshold required for C3 photosynthesis, the form of photosynthesis used by the vast majority of plants. Plant life will begin to collapse. With the collapse of plant life will come the collapse of the atmospheric oxygen replenishment that plants provide, and atmospheric oxygen will begin to decline. Over the following hundreds of millions of years, the oxygen age of Earth will quietly end. The atmospheric chemistry that was built over 2.5 billion years of cyanobacterial and plant photosynthesis will be gradually undone by the same relentless physics, the brightening of a star that ultimately enabled it. In the very longest view, Earth's atmosphere may return to something resembling its pre-oxygen state. Not identical, the geological and biological history of the intervening billions of years will leave its permanent mark on the planet's surface chemistry. But oxygen-depleted, greenhouse-dominated, hostile to the complex aerobic life that has filled the planet for the past 500 million years. This is not an emergency. 1 billion years is a duration so vast that the entire history of complex animal life on Earth would fit into it nearly 4 times over. But it is a reminder, written in stellar physics rather than biology, that the oxygen world is not the final state of this planet. It is an episode, an extraordinarily consequential and beautiful episode in a much longer story. Not as an intellectual exercise, as a genuine act of imagination. Start at the beginning of this story, in the warm, sunlit shallows of an ancient ocean on a world you would not recognize, and walk forward through every step. A cyanobacterium splits a water molecule. The oxygen drifts upward through mineral-rich water, reacts with dissolved iron, and settles to the seafloor in a rust-colored particle that will eventually become part of a banded iron formation still visible in Australian rock today. The cyanobacterium divides. Its daughters divide. Across millions of years, the process multiplies until the iron in the ancient ocean is exhausted and the oxygen begins to escape into the atmosphere above. The ancient world begins to die slowly, chemically, completely. The methane blanket dissolves, the temperature plunges, the planet freezes. In the deepest, darkest, most extreme corners of a frozen and oxygen-poisoned world, survivors persist, adapting, mutating, stumbling toward new biochemical possibilities in the only way evolution knows how to move. An archaean cell fails to digest an aerobic bacterium. The mistake becomes a partnership. The partnership deepens over millions of generations into permanent biological integration. The mitochondrion is born. Energy abundance cascades through biological possibility space. Cells grow larger, more complex, more capable. Multiple cells discover the advantages of collaboration. Bodies appear visible to the naked eye, built from trillions of coordinated cells, each one running the same aerobic metabolism that the ancient captured bacterium introduced. The Cambrian explosion fills the oceans with eyes and shells and legs, and the first nervous systems complex enough to process the information those eyes collect. Vertebrates appear animals with internal skeletons that can scale to sizes and speeds no external skeleton can match. The ozone shield thickens. The land opens. Plants colonize the continents, building soil and oxygen and the food webs that will feed everything that follows. Insects, amphibians, reptiles, the first warm-blooded animals huddling through ancient winters with their elevated metabolic rates and their increasingly complex brains. Mammals, primates, a lineage that begins to make tools, to speak, to ask questions about the past. Each step in that chain was contingent on every step that preceded it. Remove the cyanobacterium, and there is no oxygen rise. Remove the oxygen rise, and there is no mitochondrion. Remove the mitochondrion, and there is no complex cell. Remove the complex cell, and there is no animal body plan. Remove the animal body plan, and there is no nervous system. Remove the nervous system, and there is no brain. Remove the brain, and there is no one to read this story, to feel the weight of it, to sit in the dark and consider what it means. You are not a destination that history was moving toward. You are the current end of a chain of accidents so long and so specific that the probability of its occurring, calculated in advance, would approach zero by any meaningful measure. And yet here you are, at the end of the chain, breathing the oxygen that started all of it, thinking thoughts that would be impossible without every catastrophe that came before. There is a version of gratitude that is easy and comfortable. The gratitude for things you expected, things that feel like your due, things that have always been present and seem simply to be part of the natural order. And then there is a different kind of gratitude, rarer and more unsettling, that comes from genuinely understanding how unlikely and contingent the things you depend on actually are. The air you are breathing in this moment is not the natural state of a rocky planet orbiting a medium-sized star. Across the solar system, across the vast majority of planets in the observable universe, atmospheres rich in free oxygen do not exist. Venus has almost no oxygen. Mars has almost no oxygen. Every gas giant, every ice world, every airless rock in our planetary neighborhood has no significant free oxygen whatsoever. Oxygen-rich atmospheres are, as far as current astronomical evidence suggests, extraordinarily rare features of a very specific kind of world. A world that has been actively maintained by photosynthetic life for billions of years. A world where the continuous biological labor of converting sunlight into chemical energy has been running long enough to transform the entire atmospheric chemistry from its default state into something almost completely different. Remove the photosynthesis, and the oxygen disappears in geological moments. The chemistry of rock surfaces, of volcanic gases, of ocean chemistry will consume it, react with it, combine it with other elements until none remains free in the atmosphere. The default state of a rocky planet's atmosphere, in the absence of sustained biological activity of a very specific kind, does not include significant free oxygen. It never has. This means that the oxygen you are breathing right now is not a background feature of the universe. It is an ongoing biological achievement produced, as it has been for over 3 billion years, by the continuous photosynthetic activity of cyanobacteria and plants, maintained against the chemical tendency of a rocky planet to consume it, dependent on conditions that have been stable enough for that biological activity to persist. The Great Oxidation Event demonstrated what happens when biological activity changes atmospheric chemistry at a rate that outpaces the system's ability to absorb the change. It took hundreds of millions of years and cost the lives of the dominant biological communities of an entire era. The geological record contains this lesson in extraordinary detail, preserved in rust-red rock layers, in isotope ratios, in the chemical signatures of an ancient catastrophe that still shapes every aspect of the world we inhabit. We are the first organisms in Earth's history that can read that lesson. The cyanobacteria had no access to the banded iron formations building beneath them. They had no molecular clock, no isotope spectrometer, no capacity for geological inference. They could not know what their chemistry was doing to the world. We have all of these things. The record is open. The lesson is written in stone, in atoms, in the genomes of every living thing on this planet. What we build from that knowledge is the only question that remains entirely ours. There is a specific perceptual shift that happens when deep time stops being an intellectual concept and becomes something you actually feel, when the numbers stop being numbers and start being duration, real, and weighted, pressing against the edges of comprehension with a force that changes the way the present moment looks. Most of us move through our lives in a temporal frame that extends, perhaps a century in each direction. Our grandparents' memories behind us, our grandchildren's lives ahead of us, with the texture of human experience filling the space between. History stretches the frame further a few thousand years of written record, a few tens of thousands of years of archaeological evidence, the deep human past fading into a region that feels ancient but is, on the scale of Earth time, practically this morning. 2.5 billion years is something else entirely. If the entire history of Earth were compressed into a single calendar year, the Great Oxidation Event would have occurred in late February. The dinosaurs would appear on December 13th and vanish on December 26th. The entire span of recorded human history, every empire, every war, every invention, every philosophical tradition, would occupy the last 17 seconds of December 31st. Sitting with that compression, really sitting with it, not as a curiosity, but as a genuine attempt to feel what it means, produces a particular kind of vertigo that is also, strangely, a kind of liberation. The problems and concerns that fill ordinary human time, the conflicts, the anxieties, the ambitions do not disappear when held against deep time, but they become proportionally legible in a way they cannot be when viewed only from inside the human temporal frame. They are real, and they matter, and they deserve whatever attention and effort they require. And they are also the concerns of an organism that has existed for less than the last second of a year-long story, on a planet that was already ancient before the first animal drew breath. The cyanobacteria were here for billions of years before anything complex existed. They will almost certainly be here long after everything complex is gone. The banded iron formations on the seafloor were building for hundreds of millions of years before any eye existed to see them. The ozone shield overhead was constructed molecule by molecule over timescales that make civilization seem instantaneous. Deep time does not make human experience less significant. It makes it more precise, more accurately located in the vast context of what has happened before and what will happen after, more clearly understood as the extraordinary and temporary and specific achievement that it actually is. Knowing this story changes what the sky looks like. It changes what a breath feels like. It changes the way silence sounds, in the late hours of the night, when the world is still and the only thing moving is the slow, patient chemistry of the atmosphere, doing what it has been doing for 3 billion years. Go back one more time, just once more. The sky is the color of rust and amber. The oceans shimmer with the chemical iridescence of an iron-rich world that has never known anything different. The atmosphere pressing down on everything is thick and methane-laced and lethal to every living thing that will exist for the next 2.5 billion years. There are no footprints on the volcanic rock. There are no songs, no languages, no questions. The only sounds are geological wind across bare stone, the hiss of hydrothermal water meeting cold ocean, the deep subsonic tremor of a planet still cooling from its violent formation. And in the shallow, sunlit water near a volcanic margin, something unremarkable is splitting a water molecule. It does not know it is doing anything important. It does not know anything at all. It is following the chemical instructions encoded in its own structure, ancient instructions, refined through hundreds of millions of years of evolutionary iteration, that say simply, absorb light, split water, use the hydrogen, release the rest. Over and over. Every fraction of a second, every day, every year, every million years. The rest drifts upward, invisible, odorless, chemically aggressive. A byproduct, discarded without ceremony, into an ocean that absorbs it for hundreds of millions of years, until it cannot absorb it anymore. And then the world begins to change. It dies and freezes and endures. It adapts in its margins. It builds new chemistry from the wreckage of the old. It discovers that the poison can be made into power, that what destroyed the ancient kingdoms can fuel something entirely unprecedented. It fills the oceans with complexity, and the land with life, and the upper atmosphere with a shield that makes the land habitable, and eventually fills that land with organisms that develop eyes and brains, and eventually voices, and eventually the capacity to look backward through all of this and ask what happened. You are the question that the universe asked itself through 2.5 billion years of chemistry. Take a breath. A real one, slow, deliberate, aware of what it is. The oxygen entering your lungs right now was produced by photosynthetic organisms running the same fundamental chemistry as that ancient cyanobacterium in the warm, shallow water. It travelled through the atmosphere that their collective labour built over geological time. It passed through a stratospheric ozone layer assembled from their output. It arrived in your lungs courtesy of an unbroken biological chain, stretching back further than any other story on this planet. You are breathing the consequence of the largest catastrophe in Earth's history. You are thinking thoughts made possible by the energy revolution that catastrophe accidentally triggered. You are the current, temporary, extraordinary expression of a process that began with no intention and no awareness and no destination. The cyanobacteria are still out there, still splitting water, still releasing oxygen, still, after all this time, building the world. And the world, for one more moment, is breathing.